Hello student, reproductive system of Acanthocephala. We have to study the reproductive system of Acanthocephala. To start that is reproductive system, first is male reproductive system. There are two testes attached to the ligament strand and enclosed in the ligament sac. In the Acanthocephala and the Paleoacanthocephala, there occurs cement glands, 6 to 8 in number, with ducts leading into the common sperm ducts in the Acanthocephala. However, the cement glands forms a syncytial mass form, which a duct enters a cement reservoir from which in turn ducts enter the sperm duct. The secretion of the cement gland seals the everted male bursa around the female gonopore. Continuous with the ligament sac is a muscular tube, the genital sheath, which encloses the posterior ends of the sperm ducts, the cement glands, and the protonephridial canals. When present, there is a short penis which projects into the hemispherical or elongated bursa, a cavity made up of the internal body wall with proximal muscular thickening. The muscular cap, the bursa, can be everted to the exterior and is used in holding the female during copulation. Female reproductive system. This show a number of peculiarities. The ovary or ovaries, two which are visibly intact only early in life, break up into ovarian balls, each consisting of a syncytium from which oogonia are formed. These soon come to be free in the Paleoacanthocephala. The sac ruptures and they fill the body cavity. The ligament strands lead to muscular organs termed the uterine bell. Posteriorly, this opens into the uterine tube, bearing two bell pouches, which connects to the muscular uterus and a non-muscular vagina opening to the exterior. The vagina aperture is guarded by a sphincter. The uterine bell serves as a selective apparatus. Ovic larvae are taken in by the uterine bell and passed through the uterus to the vagina, and so to the exterior. Immature eggs are rejected and returned via an aperture in the ventral bell to the dorsal pseudocele. The selective mechanism in P. minutus appears to operate on eggs length. Most eggs less than 100 millimicron being immature. The uterine bell is continuous with the dorsal ligament sac in the archie and Acanthocephala. In the Paleoacanthocephala, the single dorsal strand connects to the inner side of the uterine bell. In M. dubis, X06 determination mechanism appears to operate with the 2n equal to 8 females 7 male. Copulation and fertilization. The male bursa is used for grasping the female and the penis is inserted. Grasping the female and penis is inserted into the vagina and sperms discharge. The secretion from the semen glands is the force over the whole posterior tips of the female and the gonopore securely sealed. The secretion persists for time as a hard brown copulation cap. Structure of egg cell. 4 egg membranes have been described in most species, 3 in others. In M. dubis, the outermost layers contains keratin. The third is Pas positive and made up of protein chitin. The innermost layer is proteinaceous. The eggs of some species are extremely resistant. Eggs of P. minutus retaining their infectivity for 2.5 months at 22 degrees centigrade. The life cycle are known in details for only a few species. The intermediate host are insects, larvae, beetles, cockroaches. For those parasitic in land animals and crustaceans or mollusks. For those in aquatic animals, some developmental stages of polymorphous minutus are shown. The time for hatching of the eggs varies with species, 2 hours being the earliest reported time for M. dubis. In the cockroach, eggs of Echinorinchus saginatus hatch in ostracods within 1 hour. Hatching is markedly efferate. pH is greater than 7.5 being necessary for optimal results. In molarity is greater than 0.2 is also essential. In the presence of carbon dioxide, however, hatching takes place at the lower pH. A chitinase appears to be involved in the hatching process and bile salts appears to be the chief agent activating larvae in the definitive host. Classification. 3 orders are recognized. Order first, Acanthocephala. Hooks on proboscis. Either in long rows or in a few circles. Spines lacking on trunk. Main lacunar vessels, dorsal and ventral, both dorsal and ventral ligaments. Sacs present in female semen glands, separate with yet present in males in terrestrial host. Example, Moniliformis dubis. Order second, Acanthocephala. Hooks on proboscis usually in long rows. Spines on trunk. Nuclei in hypodermis, usually fragmented. Main lacunar vessels, lateral ligament sac in females. Single semen glands, separate, mostly aquatic host. Example, Leptorhynchoids. thicatus in fish, larvae in amphipods. Order third means three order classification: Acanthocephala, Acanthocephala, Acanthocephala. Order third is Acanthocephala, hooks on proboscis, usually in a few small circle, few nuclei, large in hypodermis, main nuclei, vessels. Dorsal and ventral ligament sacs in female. Cement glands syncytial in aquatic host, example Neoechinurhynchus imidis. Several alternative classification to the above are also in use. This is the reproductive system, life cycle and classification of Acanthocephala. Okay, thank you.